one more presentation to take. Dr. Tolu Sherand. Okay, she's going to talk to us on Helicobacter pylori genome. Sorry, she's going to talk to us on culture of the Helicobacter pylori.
Good afternoon, great minds. I'm privileged to be standing before you this evening, and I want to appreciate my mentor, Professor Smith, that have tutored me for about 9 years. And I know that without collaboration with other people, some of the things I did in the course of my PhD research wouldn't have been done. We had collaborations with gastroenterologists from different parts of Nigeria, Professor Otegbayo, Professor Ndububa, Professor Oyekwere, Professor Rose Ujiagbe, and Professor Leslie Olufumilayo. So this evening, I'll be talking about culturing of H. pylori. Okay, I have the, okay. I'll be guided by this outline. I'll be talking about the clinical specimen for H. pylori culture. What are the suitable culture media that will enable the optimal growth of H. pylori? Incubation conditions. How do we isolate and identify H. pylori? If contamination or inhibition happens, what do we do? How do we troubleshoot? And I'll be sharing some of the results from Nigeria. H. pylori is a flagellated microaerophilic gram-negative spiral-shaped fastidious. When we are talking about H. pylori being fastidious, it doesn't grow like every other bacteria like staph and E. coli. It requires special conditions, special enrichment media to grow. So it's a fastidious bacterium associated with gastroenteritis, peptic ulcer, and gastric cancer. It has also been implicated in extra gastrointestinal disease, such as vitamin B12 deficiency, inflammatory bowel disease, immune thrombocytopenia and iron deficiency anemia. Why is culturing of H. pylori important? It is essential for diagnosis, antimicrobial susceptibility testing, eradication therapy, monitoring, epidemiological studies, understanding antibiotic resistance, research, personalized treatment, and quality control and assurance. Here is a table showing the different clinical specimens for the culture of H. pylori. Different specimens have been reported, like dental plague, blood, saliva stool, but the most suitable out of them all is a gastric biopsy, which has been reported to have high rate of isolation during culture. And there are required transportation medium when gastric biopsies are taken during endoscopy. And Stuart's medium have been reported, but I've worked with pathogen pylori. Some researchers have used physiological saline, but the most suitable out of them all, because they reported a very increased isolation rates using pathogen pylori transport medium. And during endoscopy, when these biopsies are taken from the patient, it is important to receive from the scoping, from the endoscopist with a sterile forceps. And this biopsy is immediately embedded in the pathogen pylori transport medium so that it's not exposed to air. And then transportation is done within 2 hours of, but from experience, we have received biopsies, and it took about 48 hours before we started culturing, and we were able to isolate H. pylori. Here is the endoscopic unit at LASUT. Professor Oyekwere taking biopsy from one of the patients. And then we have different selective medium and non-selective medium that can be used for H. pylori culture. For non-selective medium, we can use a blood agar or chocolate agar without any antibiotics introduced into the agar. The selective medium, we can use GC agar as the agar base. Columbia agar have been reported. Mueller-Eaton agar have been reported. Trypticase soya agar have been reported. Castman agar, brain heart effusion agar, Brucella agar, Skyros medium, dense medium. Using the GC agar, I have a picture of the agar on the screen, and above is the picture of the pathogen pylori transport medium. This pathogen pylori transport medium, and here is the GC agar. The manufacturer instruction says that 18 grams of this GC agar should be measured while preparing the agar, and this is measured into about 450 mils of sterile water, and a magnetic stirrer is introduced into the autoclaving bottle, and this is autoclaved the GC agar, 18 grams is introduced into the autoclaving bottle containing 450 mils of sterile water, and after incubation, a dense medium and supplements, vitamin mix, dense medium. This dense medium, here is a picture of the dense medium. It's a selective supplement that contains vancomycin, polymyxin, amphotericin B. This is meant to inhibit contamination from fungi, yeast during culture. And the vitamin mix contains nicotinamide, adenine dinucleotide, vitamins, amino acids, some enzymes, and some other nutrients. Because, like I said earlier, H. pylori is fastidious, and it requires an enrichment medium in order to grow. So the vitamin mix, horse blood or horse serum will be added, and most time the vitamin mix is usually about 7% to 10% is added into the agar so that it will enrich the medium. Vitamin mix and horse serum in a bottle of 450 mils, about 5 mils of vitamin mix, that's about 5%, and 45 mils of horse serum is introduced into this mixture in order to have a good balance of the media. And all this preparation is done under about safety cabinet level 2 in order to prevent contamination. We talk about H. pylori incubation, that H. pylori grows at microaerophilic condition, and this means that the atmospheric condition will require a 5% oxygen and about 5 to 10% carbon dioxide, 10% nitrogen, and about 85% of nitrogen. And this can be generated in our own local environment with a gas jar, although in a developed environment, it's possible to have an incubator that can produce this microaerophilic condition. So in our own local setting, we use a gas jar to create that microaerophilic condition using we use an anaerobic gas pack to create the microaerophilic condition using a gas jar. The gas jar could be a volume of 3.5 or 5 mils. So there is specification for the anaerobic gas pack. It could be a 3.5 mil specification or 5 mil specification. So depending on the volume of the gas jar, it will determine the size of the anaerobic gas pack to be used. Now the temperature, talking about the temperature, it requires a maximum temperature of 37 degrees Celsius. In our African setting, it's possible to have problems with electricity. This can result into H. pylori inhibition while we are culturing, because a 37 degrees Celsius for a minimum of 3 days is required for optimal growth of H. pylori. And the incubation is done for a period of 3 to 7 days. What are the cultural characteristics of H. pylori? Like I said earlier, they are microaerophilic, requiring about 5% oxygen, 10% carbon dioxide, and 85% nitrogen. They are fastidious, they require special enrichment medium, and this can be available through the addition of vitamin mix, horse serum into about agar base, which could be GC agar, Columbia agar, Brainerd infusion agar, Brucella agar, and then the growth is based on blood agar. That can be done, created with horse blood or horse serum, and incubation for about 5 days. We have had instances where we culture biopsies and we incubated for about 5 days. We almost lost hope on the culture, but after incubation for more than 10 days, we noticed that the growth of the H. pylori came up in the culture plate. So it's possible to incubate further in order to get H. pylori isolated. How do we isolate and identify H. pylori? H. pylori colony are small, between 0.5 to 2 mm, and they are usually translucent to yellowish colonies on horse blood agar. Then the gram reaction is gram-negative spiral-shaped bacterium. When we use our crystal violet, Lugol's iodine, and ethanol to decolorize, and then we do a counter staining with safranin, we have a gram-negative spiral-shaped reaction. The biochemical test to confirm the presence of H. pylori includes the urease, catalase, oxidase, which are positive for H. pylori. If you look at this, the urease reaction in the initial, if H. pylori is introduced, it will be like this, but if it's positive, it will give a baby pink color. Then confirmation can be done using PCR and sequencing. Contamination is usually occurs during culture of H. pylori. We could have fungal and yeast or other bacteria contaminating the plate. Inhibition of growth may occur through antibiotics. One of the biopsies that we culture, when there was no growth, we have to investigate if the patient have been on antibiotics, and the patient told us that he has been on a probiotic. So we had to instruct the patient to stay away for some time, and then another biopsy was taken, and then there was growth. So it's possible to have inhibition from previous use of antibiotics. It's important to do quality control in order to know if actually what you're isolating is H. pylori, using some H. pylori strain, control strains, P12, which is the European strain, European strain, the J99, the African strain. In case of no growth, like when Professor Revati was talking, that sometimes biopsies are cultured and no growth. What do we do? How do we troubleshoot that? Like I said earlier, sometimes there will be failure to isolate H. pylori. It doesn't mean the patient is not positive for H. pylori. You have to troubleshoot and investigate if the patient has been on antibiotics. Number two, in the process of collection and transportation to the lab, as it stayed longer than expected before the culture is done, this can inhibit the growth. Sometimes if the number of H. pylori is low, culture might become negative, but yield may be improved by prolonging incubation to about 12 days. Contamination can result if processes are not carried out aseptically. Monitoring of culture media and reagents can also be done. If the vitamin mix have been denatured or the horse serum are not obsolete, the growth of H. pylori can be inhibited. I present the results of culture from Nigeria between 2015 and 2018. That's a period of 3 years. About 492 subjects were recruited from different tertiary institutions from Nigeria, and we have their urea bed test done. 184 out of these 492 subjects were UBT positive. Culturing their biopsy, we had a recovery rate of H. pylori of about 125 isolates from these patients. And after looking at the urea bread test compared to the culture, we had an isolation rate of 67.9%. Susceptibility testing of H. pylori isolate from Nigeria, we use e-test, which was done on GC Hager using serum or horse blood with a vitamin mix to prepare the plate. And we tested for inhibition for metronidazole, tetracycline, amoxicillin, and clarithromycin. From the results, we had the highest concentration recorded from the e-test to be 256 micrograms per ml, and the lowest antibiotic concentration to be 0.016 micrograms per ml. The next slide I'm going to share, I know Professor Mafitaina may not be happy about it, because I have a metronidazole resistance to be 93% and amoxicillin to be about 42%. There have been a lot of controversy about the resistance of H. pylori to amoxicillin, and we investigated further on the cause of this, because this investigation was done in triplicates. We repeated the process 3 times before we concluded on the level of resistance. We investigated beta-lactamase production, and none of those isolates produced beta-lactamase. We also did penicillin binding protein 1A sequencing to check for frame shifts in the penicillin binding protein 1A motif, and we only noticed some alterations in the motif. Then we did an efflux pump analysis on the isolates, and statistically, the efflux pump HEFA was statistically associated with resistance of amoxicillin. Clarithromycin resistance was 39% and tetracycline 27%. Here is a picture of the plate for e-test. And I conclude that culturing of H. pylori, especially in Africa, is challenging because it is fastidious. We still have challenges with electricity to maintain our temperature. majority of the laboratories don't do H. pylori routinely. We only have it done in research centers, and we have many limited access to resources. Personnel were not trained. Only few trained personnel are trained on H. pylori culture. Equipment and reagents are costly. And all these things are challenging to the culture of H. pylori. So there is need to give priority to H. pylori research, because other infections like malaria, TB, hepatitis are prioritized in Africa. So culturing is crucial for diagnosis, antimicrobial susceptibility testing, eradication therapy, monitoring, epidemiological studies, understanding antibiotic resistance, personalized treatment research, and this is essential for the effective management of Helicobacter pylori-related diseases. Here are my references. Thank you.
Okay, thank you, Dr. Tolu, for that beautiful presentation. I guess there may be questions for you. Indicate by raising your hand.
I have a question.
Okay, Prof, go ahead, sir.
Of course, one question would be rather a small provocation, because we have heard before how often culture fails. And you say culture is essential, but you can, for clinical purpose, you could switch to molecular testing, because what is interesting is to know the antibiotic resistance. Now, two questions. You said you repeated 3 times for amoxicillin.
Not all the susceptibility testing for metronidazole, for tetracycline, for clarithromycin were done in triplicates.
But these strains,
Yes.
Stella, listen to, you have not sent out to maybe a European
We
or
did.
to or
We did.
Yamaoka?
We did.
To?
We did. We did.
And what did the others find? The same?
The same.
And who are the others?
We did in
We are
Nigeria. We did in Munich.
Wow.
Yes.
Did it, Yamaoka, you should comment this, but maybe tomorrow. The other thing, I think it's very interesting, because fact, we take this as a fact.
Yeah.
Now, what about the genome of those who have this antibiotic resistance? Are they belonging, obviously in Africa you have a more consistent, homogeneous, I believe, genome, not like in the United States, as Dr. Abner showed. So that probably you don't find in the context of the genomes. But what about, and maybe tomorrow, Professor Yamaoka, who studied also the various mutations in amoxicillin resistance. Yamaoka, what would be your explanation? Huh? Yeah, maybe you comment also for having something important to understand. I mean.
Yeah. Thank you, Prof.
Unexpectedly high. So it's there is a 40% is somewhat unexpectedly high. This is true. However, but we check, for example, even in other countries, especially in Africa, it's really high, and it's not different, because usually our test is done in Japan. You know, so at that time we have some control. Other countries, even we did it in Japan, it's really almost 0%. However, at the same time, in the same days, we do the African strains, it's really high. So it's really true. So maybe something wrong, and we found recently the PBP1, some 4 mutations. So tomorrow we will present the data about.
The dual therapy, 1% amoxicillin.
Will not work.
Yeah, yes.
No? So this has an immense importance for your country, for what could be the future treatment, because then amoxicillin is out for 60% of the people. So but maybe you should.
Is currently is very difficult to treat. Even the classical therapy is impossible, and even the amoxicillin, no. So even this data shows the even the tetracycline, there is some others. So it's really so we need some susceptibility test. It's really necessary. And if it's a genomically get it is okay, but currently, unfortunately, except for clarithromycin, so there is no definite mutations. So really we need some culture. Culture is necessary, so especially in Africa. But it's very difficult.
Can I share something, Mr. Chair? Chair's permission?
Somebody was
Almost the same data from Kenya. Catherine Mongi, my doctor student, her data, but differential susceptibility. From corpus, it is 39% amoxil resistance. From antrum, 50%. There is a difference between the different sites. But it is almost like what is presented.
I think the major problem is the abuse of antibiotics in Africa, because majority of patients, it is when their condition is complicated that they visit the hospital. They have access to all kinds of concussion that has antimicrobial agents in them. They order for overhead counter drugs. It is when their conditions are complicated that they visit the hospital. So majority of people abuse antibiotics in Africa.
Please, I have a question. For
She also mentioned it was real and it happened last year. You see, this person was taking antibiotics, so we told him keep off antibiotics. Who he did and came back. We couldn't get anything, VAC culture. Then we now asked him what happened. He said it takes a probiotic that he got from the US. So we said, okay, just keep off everything that is similar to antibiotics. And then after 4 weeks, we told him to come back. He did, and we cultured. But she was even telling me, let's throw the plate away. But I said, let's just leave it. So on the 11th day, it grew H. pylori. So what I want to suggest for us that are doing culture here in Africa, we should leave it longer, because you never know whether it is going to grow after the normal 5 to 7 days or something. So it's just a suggestion for us. Maybe it's something also we as Africans could also consider looking at, so that we do not actually, I know you are talking of treatment, if you take it that long, so that we do not actually have issues on saying that it's culture negative. Because this particular one we were talking about was actually real life, and that was what we noted. On the 11th day, it started growing. I was really amazed.
I think it's,
okay, I have a
somebody
question.
has.
Leave the culture for longer time, 6 weeks, 8 weeks on the solid media. Finally, we try this prolonged incubation in TB. 6 weeks, maybe the strain didn't grow, but you keep another 2 weeks, we have successfully grown and we did the susceptibility. I must also try this longer incubation. Thank you so much for that.
Thank
Okay, I think maybe the last question. You are raising your hand.
Yes.
you. Prof wants to ask a question.
All right. Thanks so much. I think for me, just one comment and then one question. One of the comments is in terms of the amoxicillin resistance. I think part of my study that I'm still currently just trying to do a write-up, we did the whole genome sequence and also look at the virulence factors as well among African samples, and then compare that to some other countries like the East Asian countries and the Europeans and some other countries, and even Japan and China. Now, one thing we see was one of the genes, the PBP tray. What we found in that sample, in the study, was that the resistance gene among Africans was quite high, about 21.4%, as against those in America, 1.1%, then those from Europe, 0%, and then those from Asia, 22.7%. So it is possible that we do have some genes that possibly will have predisposed Africans to having more resistance in amoxicillin. So that might be a factor. I'm still going to bring out more of the studies, but I'm just looking at the fact that we do have a resistance from amoxicillin, and it's not very surprising because it seems to be as if we have some genes that do predispose us to those resistance. beside the fact that also that we do abuse antibiotics a lot, which would have also been a contributory factor to that as well. But my question was this: why do you choose to extend for that particular patient, what prompted you to say, let me extend this culture time? What informed you? Because if you're going to have a lot of patients that are going to be coming, your culture, they're all negative, then you say, okay, let me extend. Why do you have to extend the culture for that particular patient? Why?
Well, I think the patient in question was actually my patient, and I knew how worried he was.
It was out of frustration.
Do you understand?
Because the first time I did it, nothing grew. And then he came again, and I didn't see anything. So I said, no, this can't happen. I can't waste my time. I can't waste my efforts. So I said, we should just leave this place in the incubator. We just change the gas pack and put a fresh one. So he was doing that on the 11th day. She almost said we should discard the place. I said, no, let's just skip it and see. After all, it doesn't cost us anything. So on the 11th day, I just brought it out, and I saw the tiny growth of H. pylori. I was just so happy that at least it wasn't a wasted effort. So that's why I'm suggesting that for us in Africa here, maybe we could try that in cases of where we have culture negative issue, because it's his patient. He's failed eradication therapy thrice.
He's actually a pharmacist.
Yes,
and
thrice.
He has access
so we were all
to everything.
frustrated. We wanted to see the end of the work, and that's why, and it yielded some results. And today he's completely eradicated from H. pylori.
The African Helicobacter and Microbiota study group, we made a video on the culture of H. pylori, and you can access the video on the YouTube link displayed on the screen. Thank you.
Okay, thank you so much for that presentation. I think that rounds up this session, and thank you for your audience. Thank you.